In this mini video, we are going to be talking about DNA and its structure. Watson and Crick used X-ray diffraction images that Rosalind Franklin had taken to begin building their model of DNA. Using their knowledge of chemical structures and previous research, they were able to start. When Watson discovered that bases paired in a pattern of adenine to thymine and guanine to cytosine, they were able to start building their 3D model. Adenosine and guanine have a double ring structure. This makes them purines. Cytosine, thymine, and uracil, which is only found in RNA, have a single ring structure. This makes them pyrimidines. The base pairing rules that they discovered, C and G, with A and T, helped them build their 3D structure. They began building their model with these base pairing rules in mind, and they created a ringlet of anti-parallel strands, meaning they read in opposite directions. Sugars and phosphates faced outwards, with the bases facing inwards. Each strand reads 3 to 5 prime, and with their base pairing rules, they are anti-parallel. The three-dimensional structure of DNA presents as a double helix oftentimes, and within that double helix, the structure is composed of hydrogen bonds and a phosphate backbone. The hydrogen bonds connect complementary bases of different strands, while the stacking of the base pairs also stabilizes the molecule. The phosphate backbone provides phosphodiester bonds that connect the 5' phosphate group and the 3' hydroxy group of neighboring nucleotides to the same strand. The base attaches to the 1' spot on the deoxyribose ring, and the phosphate attaches to the 5' position. 2' holds the H's and the 3' the OH. Ribose for RNA is almost exactly the same, except for the 2' spot has an additional OH instead of just an H. To tell if a helix is left or right-handed, I imagine looking down at the coil from above, and the direction of its coil indicates whether it is left or right-handed. A major groove is where the helix backbones are spread apart, while the minor groove is where the backbones are close together. Keeping all of this information in mind, what if we were to put DNA in a concentrated salt solution? What would happen to the temperature melting? The melting temperature is the temperature at which the DNA decompensates. The lower the salt concentration, the lower the temperature at which the DNA will melt. In high concentrations of salt, DNA is essentially buffered by the negative salt ions, and the heat is unable to penetrate or degrade.